So I will try to, since I'm what at the Tour de France you would call the la voiture balai, or the broom car, I will try to be short and hopefully efficient. What I will go through is, we'll get back to commensals a little bit, but I'm not sure I'm ready to talk too much about commensals. What I would like to talk more about is what is actually in the title, is sort of how the host deals with commensals versus pathogens, and also get back a little bit on the definition of pathogens regarding the fact that they are not just microbes that induce very strong innate immune reaction in the host, but also regulates this immune reaction. And in a way, the message that it conveys, and I think we all agree once working on pathogens, is that some of these effectors which are regulating, for instance, the innate immune response, might turn out to be extremely interesting in terms of identifying new targets to develop new antimicrobial drugs, new anti-inflammatory molecules, and so on. So the idea is to let the bacteria or the microbes educate us on the good targets. So I will speak, of course, about Shigella at some point, even though it is not the best commensal that you could dream of. But anyway, so the first slide is something which I think has been a recurrent theme during this meeting, which is the fact that, and this has really been, I would say, a sort of revolution over the last, let's say, 5 years, that you are all acquiring some sort of a global view of microbial interactions with the host. And this is, I would consider that this is certainly a progress. It's been helped a lot by technological developments, especially metagenomics, as we've heard from different people here, including Dushko this afternoon. So there has been a change of scale in the way we are looking at things, but at the end of the day, I think there will be also a need for the old-fashioned guys like us, or at least like me, who try to get into the sort of details of the molecular interactions. And the message here is that, again, global means also combination of approaches. So we have the commensals on the one hand, huge numbers, especially in the gut, particularly in the colon, and we've heard a lot over the last 2 days about how the signaling that is induced, especially at the epithelial surface, is sort of filtered and translated into tolerogenic processes, and how ruptures, disruptions in these mechanisms of tolerance may need one way or another, let's again be global, to diseases like inflammatory bowel diseases, like metabolic diseases, and so on and so forth. We are even now getting to possible alteration in neurological developments, behavior, and so on. So this is one thing. And on the other hand, of course, we have the pathogens, especially the bacterial pathogens, which are hitting the epithelium very hard, which are inducing a very, very strong inflammatory reaction, which is essentially dedicated to the destruction of these microbes. And most of the destruction that we see during infection are actually due to the innate immune response more than to the microbe itself. But the big question mark, of course, is always, how with the similar systems of microbial sensing, the toll-like receptors, the nod-like receptors, to remain within the limit of bacteria, how do we, as hosts, discriminate between the symbionts and the pathogens? And this is really a recurrent question, which might not be so easy, actually, to solve. So what I would like to say here is that if you are a bacterium, and if you want to sort of make your life out of this very complex ecosystem that is represented by the intestinal epithelium, you have this mucus layer that you've heard about, several talks and beautiful new results. The bacteria are actually sort of engaging this mucus layer. The problem is that this mucus layer also certainly, and we've discussed that as well, serves as a sort of matrix for antimicrobial molecules like antimicrobial peptides, cationic peptides like this beta-defensin 3. This is extracted from a work from Brice Perandio and Thierry Pedron in the lab, showing that at the surface of the epithelium, this brownish layer here corresponds to an impregnation of the deepest mucus layer by these antimicrobial molecules. So it kind of materializes the situation that microbes have to face when they venture in this very dangerous area. So if they want to survive not only these antimicrobial peptides, but other factors like NO, RAS, lysozymes, proteases, cite them. I mean, there are lots of products which are very deleterious, bactericidal for these microorganisms, not even speaking about transmigrating phagocytes. You have to combine strategies. One is survival, and the other one is, again, if you venture more deeper into this ecosystem, subversion of these host immune responses. Otherwise, you will never make it. So, as a symbiont, it's probably the good strategy is probably to stay at respectable distance from the intestinal epithelium, and that's probably more or less what we see, even if it's sort of a naive view of the situation. We have the intestinal epithelium here, we have the mucous layer, we have all these defense sensing and mechanistic systems to degrade the microorganisms. And so the bacteria stay away from this, it's been called demilitarized zone, you can call it no bacterial land or whatever you like, but they remain at good distance. And between these two populations, in the epithelial cells and this complex microbiota, there is this situation of physiological inflammation, which is established. The bacteria speak to the cell respond to the bacteria, and there is a balance somewhere which is established, which of course can be disrupted at any moment. So that's more the sort of situation which is leading to this tolerogenic cascade of signaling. For the pathogen, it's a completely different situation because these pathogens are poised to sort of engage the intestinal epithelium or any other epithelia, and this is inherited in their genome. I mean, you see this pathogenicity island and bacteriophages encoding elements like mucinases, adenosines, invasins, type 3, type 4 secretion system that inject effector molecules into the cells. And this is essentially leading to a disruption of this tolerogenic cascade as well as induction of a very, very strong bona fide innate immune response that comprises also, again, the attraction of these transmigrating phagocytic cells. So we are really in a sort of, this could be considered the two poles or the two extremes of a spectrum, and in between, of course, we have these now called pathogens, SFB, Helicobacter hepaticus, and probably others, which are playing on the two sides in a way, depending upon their quantity, depending upon their location. They are causing lesions sometimes, or they are helping the system to get established by putting pressure, especially on the innate immune system. So these are very, very fascinating microorganisms in a way. So in terms of the differential signaling, I mean, it's true that the commensals, the symbionts, are sensed by the tolls, by the non-like receptors. So what makes the difference? I mean, we are more and more getting into this concept that it's kind of two level of signaling. In addition to the classical sensing of the PAMPs or MEMPs, we now have this notion of danger signal, which is sort of put on the epithelium, on the cells, epithelial cells of the immune system, damage to the membrane, strong adhesion, invasion, penetration of the bacteria into the cells, release of some PAMPs like flagellin, for instance, in the intracellular compartment, or release of peptidoglycan fragments in the intracellular compartments. All these things are sort of signals that something wrong is occurring, that the epithelium is strongly engaged by the microorganisms, and it's not just the bacteria sort of established somewhere at a respectable distance, as I said before. So this is a sort of a novel view, I would say, in general, of the discrimination process, which was actually nicely illustrated by this review from Vance and collaborators, and which we are also try to contribute over the years. So I would like to take Shigella as a model of disruption of all these processes, but also as a model of regulation of the innate immune response. So Shigella has this type 3 secretion system, which I already mentioned, which is necessary at all stages of the pathogenic process to cross the follicular associated epithelium, to kill the macrophages, the resident macrophages by pyroptosis, to penetrate, invade into epithelial cells, escape in the cytoplasm, facilitates the migration from one cell to another, and so on and so forth. So that's the sort of cell biology game that we've learned over the years. But what I would like to focus on here is the fact that as the bacteria engage epithelial cells, they program or reprogram the epithelial cells to become pro-inflammatory. So there is a strong correlation between the expression of this invasive phenotype, especially the expression of the type 3 secretion system, and some of the molecules which allow the bacteria to engage the membrane, alter the membrane, and penetrate into the cells, and the activation of pro-inflammatory genes, like genes encoding pro-inflammatory cytokines, chemokines like interleukin-8, which attracts polymorphs, CCL20, which attracts dendritic cells, and so on. Also, of course, genes encoding the antimicrobial peptides, the ones at least that can be induced, they are induced in this pro-inflammatory cascade. So this is really leading to a sort of disruption of the epithelial barrier by the group of cells which are immigrating and disrupting, and the bacteria can make it in this very complex barrier. But there is, of course, a limitation to this, which is that if inflammation is elicited, of course, the main purpose of this inflammatory response is to kill the microorganism. So one way or another, the bacterium needs, and I think it's written somewhere, the bacterium needs to be able to downregulate this innate immune response to be able to survive and efficiently colonize and invade. So it's kind of that which I mentioned in my title, the yin and the yang. You push on the inflammation because it's necessary to make your way through the epithelium, but at the same time you have, or maybe a little delayed, you have to control this inflammatory response. So that's an important point. But the other point is that as we've been progressing in trying to understand the pathogenesis, we figured out that we had missed one thing, which is the fact that the bacteria not only interact with the epithelium with all the major consequences that it has, considering the fact that the epithelium is really the first line of interaction with the microbes, but also to the immune systems and cells of the immune system, and this will be the second part of my talk. So just to make a very long story short, I would just like to mention very quickly the work of Benoit Martin, because this is, to me, illustrating how the bacteria behave when they live in the intestinal lumen, starting to engage the intestinal epithelium and how they can adapt. And just to summarize very quickly, this was published last year, it turns out that Shigella very nicely adapts its pathogenic capacity, especially the efficiency or the competence of the type 3 secretion system according to the oxygen concentration. So when the bacterium is in the anaerobic conditions that is prevalent in the intestinal lumen, the type 3 secretion system cannot be activated. Therefore, the bacterium keeps this pool of effector molecules. It has very long needles because it doesn't change this sort of switching process from synthesizing needles and putting together the apparatus at the tip, which is necessary to engage the epithelial cells. But as soon as the bacteria reach the surface of the epithelium, their type 3 secretion system becomes competent and it can be activated, and the bacteria can efficiently engage the epithelial cells. And what is governing this sensing of environmental condition is a gene which is called FNR, and this is a work in collaboration with the group of Christophe Tang at Imperial College, which is the gene that regulates the adaptation of the bacterium to anaerobiosis. And it turns out that this gene has been harnessed by the type 3 secretion system operons to govern the competence of for secretion. So that didn't make so much sense because, again, the situation is a situation of anaerobiosis, but as it turned out in this experiment, which is an in vivo experiment in the rabbit, this is the intestinal epithelium here, that's the lumen. Red is for the bacteria, these are E. coli which express GFP, but what you can see is that only the bacteria that are here sitting at the surface of the epithelium have fluorescence expressed, and this is because GFP is dependent on oxygen. So this is essentially, as I would say, mapping the oxygen presence at the surface of the epithelium, and then it goes dark here because there is no more oxygen here, it's all anaerobic. So basically the bacteria adapt their type 3 secretion system competence through the fact that oxygen retrodiffuses from the mucosal surface here into the lumen and become competent for invasion. And we are now working, of course, on trying to figure out how much the oxygen is present and whether it's, for instance, supported or embedded as antimicrobial peptides, for instance, in the mucus layer. So this is, in a way, a new view of infection depending upon oxygen concentrations. So as Shigella engages the epithelial cells, it of course causes this reprogramming of the NF-kappa B pathway, for instance. And 10 years ago, with Dana Philpott and Stéphane Girardin in the lab, we could demonstrate, of course, that the NOD1 and NOD2 molecules were actually sensing fragments of the neuropeptide of the bacterial cell wall, and through this cascade that goes through RIP2 could engage the activation of the pro-inflammatory genes, the NF-kappa B and the MAP kinase pathway. So this is sort of a global view of the situation. But what we learned from the work on Shigella over the years is that there are lots of effectors which are produced by Shigella, which are injected by the type 3 secretion system, that are able to downregulate this very strong inflammatory reaction. Some of them are part of the pool of proteins that are constitutively expressed at 37 degrees, and others are actually part of this regulon, which we call the MXIE regulon, which has been dissected by Claude Parceau in the lab over the years, which sort of couples the sensing by the bacterium of the activation of the type 3 secretion system and the transcription of these genes like OSPG, partially OSPF, and all these EPA-H molecules. So if we go throughout the list, the first conclusion is that all these effector molecules that are injected by Shigella into epithelial cells are actually enzymes, so it brings this catalytic dimension to the signaling process. IPGD, which is part of this pool here, constitutively expressed at 37 degrees, is a phosphatidylinositol phosphatase, which hydrolyzes PI45P2 into PI5P. And this has been shown over the years by different people in the lab, but more recently, Andrea Puer has shown that this is a very potently anti-inflammatory molecule, and I will come back to this very quickly in a second. OSPG is a kinase which essentially blocks the function of the I-kappa B molecule and blocks the activation of NF-kappa B by hampering the translocation of the NF-kappa B transcription factors into the nucleus. OSPF, as has been shown by Laurence Arbib in the lab, is a protein that dephosphorylates the MAP kinase. It's translocated into the nucleus and essentially acts in a very complex cascade. It docks to chromatin active areas for the transcription of pro-inflammatory genes. And from there, it essentially dephosphorylates the MAP kinase and leads also to the dephosphorylation of one histone, which is histone H3, and this basically imposes sort of epigenetic regulation of the pro-inflammatory genes, especially on the promoters like interleukin-8, for instance. So these are all published data. I just wanted to put them in perspective. And EPA-H's are compose a new family of ubiquitin ligases for which we are looking for substrates. So just to summarize several years of work, I would say that there are several ways by which these effector molecules, single molecules or the molecules all together as a cocktail, are downregulating the innate immune response. They suppress, as was shown by Brice and Thierry, the expression of antimicrobial peptides. They suppress the transmigration or recruitment or transmigration, for instance, of polymorphonuclear cells, and this is very much the function of the OSPF molecules. And again, as I quickly signaled, they can also block dangerous signaling, and this is the work of Andrea, showing that phosphatidylinositol phosphatase degradation of PI45P2 is actually leading to the closing of these hemichannels, thereby blocking the release of ATP, which is a dangerous signal. So it's just like the bacterium is making the epithelial cells unable to alert adjacent cells in a paracrine pathway, and this is a work that is going on in collaboration with Guy Tranvagnieux, who is here now at the Collège de France. So there are several ways by which the epithelial cell signaling can be manipulated, and this is just a quick survey, and these are the people involved in some of these important steps. The last part of my presentation, I would like to get to something which we had been discussing a lot in the lab over the years, especially with Armel Falipon and the people working with Armel in the lab. A major observation in the case of Shigella is that the immune response is very inefficient. And to make a long story short, it turns out that for an intracellular pathogen, or at least a bacterium that spends a good part of its time inside the cytoplasmic compartment of cells, the T cell response is very poor, very, the weak, and the immune memory, and this was shown by a work of Gernot Selge in the lab recently and published in Journal of Immunology. Poor immunological memory, something you would say the bacterium has probably a means to directly interfere with this T cell response in order to dampen this adaptive immune response. So we started to think that, the bacterium not only engaged the epithelium, it also engages areas like Peyer's patches, or at least lymphoid structure associated with the colonic mucosa, which is the major target of Shigella infection. And also in terms of immune response, we know that the bacteria cross this barrier and they collect also into mesenteric lymph nodes. So it makes sense that the bacteria may actually directly engage cells in lymphoid structures, and of course T cells would be one of the major targets. So again, this is work with Armel and a very talented graduate student from Germany, Christoph Conrad, and another very talented postdoc from the United States, Vilmara Salgado-Pabon. And I will go quickly through this data and I will close. So what we have shown actually is that the bacteria in the Peyer's patches in a rabbit model of intestinal infection are associated with T cells, and this is very significant in terms of immunostaining. And then, Vilmara started, we acquired a 2-photon microscope, and the idea was to look at, in an immunological context, in a lymph node, how the bacteria could affect the dynamics of the T cells, and on the longer term, of course, the dynamics of T cell interactions with antigen-presenting cells, for instance. So what she has done is to establish conditions in perfused popliteal lymph nodes in the mouse, where you inject the bacterium in the food pad, and then the bacterium go through the lymphatics. They get collected in this popliteal lymph node. You take this lymph node out, perfuse it in proper conditions, and you start looking with the 2-photon microscopes how T cells move and behave in a way, especially in this cortical sinus, which turns out to be a very critical area where, of course, the bacteria are also collected. So she started to look at this, and you, of course, did the classical sections that you can do with a 2-photon microscope using mice in which T cells could be loaded with dyes and then re-injected, or mice that would, by transgenesis, express GFP in some T cell populations, and started to look at the dynamics of T cells. So these would be T cells, and we've been educated to do all these experiments by Philippe Bousseau on the campus at Institut Pasteur. These are, the red are bacteria, the blue is the second harmonic, which shows the collagen fibers, and these are the T cells moving, and you can basically draw the sort of trajectories of these T cells. And of course, you can look at parameters. One parameter is velocity, the speed at which the T cells move in these systems, the confinement, how far they go, and the arrest, how long they stop moving. And when you look at these results, and this is a huge amount of work over the last 2 years from Vilmara, you compare non-infected lymph nodes, you compare lymph nodes infected with a non-invasive Shigella type 3 secretion system knockout and the wild type. And you see that for all the criteria, at least these two, there is a strong decrease in velocity, strong decrease in confinement, and on the other hand, a high rate of arrest of these cells, which do not move as much as the ones in non-infected or infected by non-invasive microorganisms. So this is with these OT2 cells, which are fluorescent. You see a non-infected situation. This is sort of materializing the trajectories. Now, if you look at the infected situation with a wild-type microorganism, still lots of cells move, but you might see some of them which are associated with microbes. And just to focus, what Wilmer did was to sort of put frames around the ones that didn't move, and this is shown here. And what it's shown is that the ones that don't move are essentially the ones that are associated with microorganisms, either on their surface or already engulfed inside the cell. So the global idea is that the fact that T cells are engaged in the cortex of these, the sinus cortex of these lymph nodes by Shigella is blocking their motility, which is, of course, going to have a strong impact on the immune response or the T cell response. So here comes, I think I changed the chronological order a little bit just for the sake of the presentation, the work by Christoph, who basically, and this is in collaboration with Elisabetta Frimiglica and Katrina Notolfer, got together on this very nice project. This is showing different things. One is that, again, in vitro, as you put a gradient of motility with a chemoattractant, activated T cells are engaged and invaded by Shigella, whereas the non-activated T cells are not. So this was a very important issue, which was, a sort of critical in the thinking we had about the whole system. But then what was shown is that the blocking in the motility process, and the blocking in motility is largely due to the loss of polarity of the movement in this case, is essentially linked to the expression of one protein, which is this effector molecule IPGD, the phosphatidylinositol phosphatase that I was mentioning. So you see here wild-type microorganisms that has engaged these T cells in this in vitro assay of motility, and this is the mutant, and this is the control. So we have really a system in which one single molecule accounts for the slowing down and blocking of the motility of T cells. So again, as I said, this phosphatidylinositol is transforming PI45P2 into PI5P, and there is a pool of PI45P2 in lymphocytes, which is associated with the membrane and linked to the dynamics of the cytoskeleton and basically controls the motility and polarization of these cells. And what is shown in these experiments here is that actually this pool of PI45P2 is essentially disappearing when the cells are infected by microorganisms, which produces the IPGD molecule, whereas if you do inject a molecule which is a mutant enzyme of this phosphatidylinositol phosphatase, you keep the pool of phosphatidylinositol at the periphery. So this is really a very nice system of interaction. And there is literature about motility and polarity of lymphocytes, which has emphasized the role of ERM molecules, especially ezrin, radixin, moisin, but especially ezrin. And these molecules are actually activated. They are phosphorylated to sort of engage the cytoskeleton and link outside receptors to this subcortical cytoskeleton. So this phosphorylation that is required for ezrin to be efficient at coupling the receptors to the cytoskeleton here is linked to the presence of PI45P2. So if you decrease or hydrolyze this pool of PI45P2 at the membrane, you basically block this phenomenon, and that's essentially what happens in the system. And by doing this, you essentially block the capacity of the cell in the presence of a chemoattracting gradient to polarize its receptor and move forward. So this is just sort of biochemical confirmation that The wild-type strain, Shigella, when it engages the cells, essentially blocks the phosphorylation of ezrin, whereas the delta IPGD mutant here doesn't block the phosphorylation process and let this process essentially go on in terms of motility. So we are really in an interesting situation now where we have evidence on the one hand that the bacterium is able to manipulate the epithelial cells, and through this manipulation of epithelial cells, of course, impact on the subsequent events of innate immune response. But we also have now clear evidence through this work that the bacteria can directly engage T cells, especially activated T cells, in the context of lymphoid structures where they are and where they see the bacteria, as you've been able to see, especially in the two-photon microscopy experiment, and essentially block the capacity of these cells to move forward and attain their targets wherever it's necessary. And again, I mean, there are effectors, there are enzymatic activity. You can start to really pinpoint to the very detailed mechanisms of interaction. And what is very interesting in this system is that it doesn't necessarily take that the bacteria are invading the T cells. It's just that the bacteria can bind to the surface of the T cells, inject their effector molecules, especially IPGD, and block the motility and pass to another cells and so on and so forth. So it's a pretty efficient system for the bacteria to disconnect the capacity for the host to mount its adaptive immune response. So this is going back to this slide that has been kindly shown by a couple of colleagues. But while we think about commensals, we think again in terms of establishing a balance that leads to tolerance of these microorganisms. When we think in terms of pathogens, we think more in terms of massive disruption of these tolerogenic mechanisms and induction of very, very strong inflammatory response. The truth in general is somewhere in between, because this is where coevolution has led. It has led to a sort of deal between the host and the pathogenic microbe, where the host can survive and the microbe also can survive. Otherwise, it's the end of us. And this is achieved by the capacity for some of these pathogens, at least model pathogens like Shigella, to inject effector molecules not only in the epithelium but also in immune cells and basically interfere with the dynamics, the activation of these cells to sort of tip the balance to a certain level. And what is really key now is to establish the proper kinetics in vivo to be able to show that to a certain phenotype of cell dysfunction correspond the injection of some effector molecules. So there is a big challenge ahead to try to make sense out of all this. And of course, at some point it will come back to understanding the mechanisms of symbiosis or commensalisms, because we are more and more interested in seeing how microbes, microbial products actually affect the dynamics of epithelial repair processes, for instance, or restitution as it's called. And it's clear that pathogens will at some point impact on this process. We used to look very much at the inflammation. I think we should look more and more, and this is to me also part of the innate immune concept, at the mechanisms of tissue repair. So I will stop here, just mentioning the institution that are still tolerating me, this support, and the people who have been participating to this work. I think I mentioned them as I was showing the results. Our collaborators, especially in this case, Philippe Bousseau, who has been of great help to Vilmara and Armel to set up these experimental conditions with the two-photon microscope, and also the group of Andrés Alcover, who is involved in this T-cell motility processes, and our colleagues, especially my good friend Guitran Vanier, who is now here with his group at the Collège de France, working on the very fine mechanisms of microbial cell interaction. And with this, thank you very much again.